Hello everyone. In this video, let us learn auxanography. Now, auxanography is a primary screening technique which is largely employed for detecting microorganisms which are able to produce growth factors such as amino acids and vitamins extracellularly. So, auxanography is to detect the growth factor producing microorganisms from a heterogeneous population. So, let us see how to perform this technique. So in this technique there we have to prepare two plates. So it is done in two steps. So let us see the steps involved in primary screening. The first step is the preparation of first plate. So let us look into the preparation of first plate. Here we are going to take a petri plate. Then we also take a filter paper strip which is about 1.5 into 2 centimeters long, and this filter paper strip is put across the bottom of the petri dish in such a way that the two ends pass over the edge of the dish. Later, a filter paper disc of a petri dish size is placed over a filter paper strip. Another filter paper disc is taken and it is placed over a petri dish on the filter paper strip. On the filter paper strip, we are going to keep a filter paper disc. Later, a nutrient agar which is about 45 degrees Celsius is poured on the paper disc in the dish and it is allowed to solidify. Later, a microbial source material such as soil is subjected to the cereal dilution. Later, the aliquots of the cereal dilution is plated on the nutrient agar, on a solidified nutrient agar, which will produce well isolated colonies on incubation. Upon incubation, these aliquots that we have inoculated by spread plating, the soil aliquots that we have inoculated by spread plating will produce a well isolated colonies after incubation. So this is the preparation of first plate. Now we will look into the preparation of a second plate. In the second plate, a minimal medium which is lacking the growth factor which is required for the growth of the test organism. So when we say minimal media is a media which will lack one growth factor which is important for the growth of a particular organism. So here we are going to take a test organism. This test organism will require, for example, I am talking about an amino acid here. I'll take an example of an amino acid. For example, I've prepared a minimal media which will not have histidine in it. Now this histidine is a very important growth factor for the test organism to grow. So I'll prepare a minimal media which will lack histidine, which will not have histidine in it. It will have all other factors except for the histidine. So I have chosen an organism which should have histidine for its growth. So like this, I will prepare a minimal media. And my test organism requires histidine, but in the minimal media there is no histidine. So I'll prepare the minimal media and I will inoculate my minimal media with the test organism. Since histidine is not present, the test organism will not grow on this minimal media. So that is how I have prepared the second plate. Now in the next step, what I will do is, now on the nutrient agar plate, from the first plate we can see that there are organisms growing. So what I will do, I will use tweezers to hold the ends of this filter paper, and I will lift the nutrient agar from the filter paper from the first plate. I'm going to lift using a tweezers. I'm going to hold the filter paper ends and I will lift the nutrient agar along with the filter paper disc. It comes out from the petri plate and I'm going to place this nutrient agar on the minimal media. You can see I have placed the nutrient agar from the first plate onto the second plate which is containing the nutrient agar. Say for example, if my soil aliquots that I have incubated, in that there are microorganisms which are producing histidine, then the histidine will seep into the minimal media. If the organism from on the nutrient agar is producing histidine, that will be my primary isolate. If my primary isolate is producing the histidine, if the organism that is there on the nutrient agar, if it is producing histidine, then that histidine will seep into the minimal media. So wherever the histidine has seeped, there the colony will grow. So wherever I can see the growth of my test organism on the minimal media, exactly opposite to that there will be a colony which is producing histidine. So I will consider that colony as my primary isolate for the production of the amino acid histidine. So like this, I can do a detection test wherein I can find which colony is actually producing histidine. So on a nutrient agar, there is a colony which is producing histidine. Will that histidine which is produced extracellularly will seep into the minimal media and it will allow the test organism to grow. So I will pick that particular colony and I'll go for secondary screening to see if it is actually helpful to go for a large scale production. So can I produce more amount of histidine amino acid or other amino acids for the production of for producing it in large scale will be checked in secondary screening. Now here I have taken an example of histidine. It could be any other amino acids or vitamins as well. So auxanography is a technique which is used to detect the amino acid producers or vitamin producers or other growth factor producers. So here we'll prepare nutrient agar plate and we'll prepare minimum media plate, and in this way we are going to see whether it is actually producing the amino acid or not. And in secondary screening we can go for other other techniques we do, other experiments we do to see whether it is really producing the amino acid in large scale. Thank you all. In my further video we'll look into enrichment culture technique and use of indicator dyes. Thank you.